Hi, my name is Will Chou. I'm the CEO of Aruvant Sciences, and I'm going to be talking to you today about our lead product, ARU-1801. So ARU-1801 is a potentially curative differentiated gene therapy for sickle cell disease. This product uses lower doses of chemotherapy than other gene therapies currently in development for sickle cell disease. The data that I'll show you today shows that ARU-1801 has the potential to be a cure with meaningful reductions in vaso-occlusive events and durable responses out 3 years. The patients that we treat, patients with severe sickle cell disease, need a curative therapy, but most importantly, they need a potentially curative therapy that they are not afraid to get. And we know that high intensity chemotherapy remains a barrier for uptake of potentially curative therapies. To take a complex product such as gene therapy from idea to full fruition requires an experienced leadership team, and that's what we have here at Aruvant. So my background is I'm a physician, and I spent the last 5 years at Novartis with Kymriah, where I led the global commercial development of Kymriah. I led the lymphoma clinical development program for Kymriah as well. And we're also led by Joe McIntosh, our Chief Medical Officer, who has 16 years of clinical development leadership and most recently led clinical development at PTC Therapeutics. Palani Palaniappan is our Chief Technology Officer. He was the global head of tech ops at Sarepta. Blair Clark-Shabe is our SVP of communications with multiple years of experience in public company communications. And Dr. Poonam Malik is our key scientific advisor, and she's the inventor of this technology and a leading world expert in lentiviral gene therapy, stem cell biology, and just general clinical care of hemoglobinopathies. So what is ARU-1801? ARU-1801 uses a third-generation self-inactivating lentiviral vector that contains a proprietary gamma globin gene for a novel, highly potent variant of fetal hemoglobin, which is hemoglobin F G16D. And the unique potency of ARU-1801 allows it to engraft with only reduced intensity conditioning. And I'm going to share with you the benefits of reduced intensity conditioning. As I mentioned before, we've shown the potential to be a curative therapy with VOE reductions from 85 to 100% and durable engraftment out over 3 years. So how do we make ARU-1801? Well, we use a patient's own stem cells to make fetal hemoglobin. First, we produce our lentiviral vector, then we collect CD34 positive stem cells, we transduce them ex vivo, and then we infuse them after a single dose of reduced intensity conditioning. Reduced intensity conditioning, what does that mean? Well, we use for ARU-1801 melphalan 140 milligrams per meter squared. So that's a reduced intensity conditioning regimen. Other gene therapies in development use fully myoablative busulfan, and you see that on the left. So what does that mean for patients? Well, there's quite a difference in time of neutropenia and thrombocytopenia. So 20 to 28 days for myeloablative therapies versus 7 to 8 days with reduced intensity conditioning. Seizure prophylaxis is required. With busulfan, the risk of ovarian failure is 70 to 80%, whereas it's 30 to 40% with melphalan RIC. So in addition to these short-term and long-term differences in toxicities, there's also differences in logistics. So the administration of busulfan requires 4 sequential days of administration with daily PK monitoring, whereas melphalan is 1 hour infusion. Busulfan requires a long hospitalization, so 4 to 6 weeks, whereas melphalan is commonly used for autologous stem cell transplant for multiple myeloma, and many of these transplants are done in the outpatient setting. So this data is from the myeloma literature, and days in hospital is anywhere from 0 to 5 days. So with our product, there is a high potential to be able to deliver this gene therapy to sickle cell patients in the outpatient setting. Now, we've talked a lot about some short-term and long-term toxicities, as well as logistics benefits of reduced intensity conditioning. But clearly what is on people's mind is risk of cancer. And while we don't have good head-to-head data, particularly in patients with sickle cell disease, of melphalan versus busulfan, we I can't provide that data because it really doesn't exist. The best we can do is provide some corollary data from the oncology literature. And so this literature says that alkylating agents, the higher dose of an alkylating agent that you give, it could lead to a higher risk of leukemia. So this is patients, 1,789 patients who were treated, they had ovarian cancer and they were treated with an alkylating agent chemotherapy. And both melphalan and busulfan are alkylating agents. We just give a dose. These patients were scored. The amount of alkylating agent they received was scored and categorized into a low dose, a medium dose, or a high dose. And over a long duration of observation, cumulative risk of acute non-lymphocytic leukemia and pre-leukemia was measured. And what you can see is at the low doses of alkylating agents, the cumulative risk of leukemia and pre-leukemia is markedly lower than at medium and high doses. And so this is the take-home point I would say from this is not to take any of this data and put it onto sickle cell disease, because this is a different condition. This is ovarian cancer. What I will say is from this data, the higher dose of an alkylating agent that you give, the more likely it is that the patient will get leukemia or MDS in the long term. So how about experience with melphalan itself? So as I mentioned before, melphalan is commonly used for autologous stem cell transplant and for multiple myeloma. And this is a study of 3,000 multiple myeloma patients who received at least one MEL-based regimen plus an auto transplant. And they were followed for up to 15 years. And you can see the different melphalan regimens included: MEL 140, MEL 200, MEL 140 plus total body radiation, beam therapy. Of note, MEL 140 was the lowest intensity regimen included in all of these different regimens that were used. And overall, this follow-up, only the incidence of overt MDS was only 0.7%, and the incidence of overt AML was only 0.2%. So it was relatively infrequent, but a known risk of melphalan. So how is it that our gene therapy, ARU-1801, is able to engraft with only reduced intensity conditioning? Well, it's three unique features of our product. First, we encode for a gamma globin with a proprietary G16D point mutation that drives a higher hemoglobin F payload per vector copy. Second, our payload itself, hemoglobin F G16D, has a more potent anti-sickling effect than endogenous unmodified hemoglobin F. And third, we have a proprietary manufacturing component that preserves the stemness of our stem cells. When you put stem cells into culture, they want to start differentiating. This additive keeps true stem cells. And it's these three features together that allow us to get durable efficacy and engraftment with only reduced intensity conditioning. So a little bit of our preclinical data on each of these effects. So the G16D mutation in the gamma globin changes a glycine at position 16 to aspartic acid. And because aspartic acid is more negatively charged, our gamma globin is more likely to bind to the positively charged alpha globin. And therefore, the gamma globin G16D is more likely to form hemoglobin F. So that was the theory for this single point mutation. And that theory actually has proven true in these well-established mouse models of sickle cell disease. So when mice were transplanted either with G16D gamma globin or with endogenous unmodified gamma globin, you can see that they created 1.5 to 2 times more hemoglobin F per vector copy in these mouse models when they were transduced with a vector encoding for modified G16D gamma globin. So the G16D gamma globin, it just outcompetes endogenous, and you create more hemoglobin F per vector copy. The second effect is the hemoglobin F G16D itself may have a more potent anti-sickling effect than endogenous hemoglobin F. So the y-axis here is reticulocytes, and lower reticulocytes means lower hemolysis, so that is good. And the x-axis is percent hemoglobin F. And the dark red diamonds are mice who were transplanted with stem cells that were transduced with vector encoding modified G16D gamma globin. So they're hemoglobin F G16D mice down here. And then in the light pink is mice who had endogenous hemoglobin F, so unmodified hemoglobin F. And as you can see, at similar levels of hemoglobin F, 30% hemoglobin F, the G16D hemoglobin F mice had lower levels of reticulocytes than those with unmodified hemoglobin F at that same percentage, 30% hemoglobin F. And so this is evidence of a more potent anti-sickling effect for hemoglobin F G16D. So next, I'm going to go through some of the data that we presented this year at ASH. Before I get into the efficacy, it's important to put the data that I'll show you into context. So we are currently in the second version of our manufacturing process. The first two patients treated were treated with process one, an academic manufacturing process. All of our processes have the G16D mutation and the stemness enhancer. These two things are what make the product the product. But before we treated our third patient, we did a few things to improve our manufacturing. We optimized our apheresis, our collection parameters. We optimized our MOI, and we also optimized the purity of the academic vector that we were using. And our vector copy number, that's the amount of vector per cell in our product, was 0.33, which is rather modest for the first two patients. For process two, we moved up to a target with these improvements, a target VCN of 1. And we're not finished there. We would like to improve this VCN even more, and we will be doing that when we roll out process 3 later on this year. We have some good evidence ex vivo that we can further increase this target vector copy number, and simultaneously, we're developing our commercial process for the start of our pivotal trial in 2022. So let's go through some of the data that we shared. So this is patient one who, again, had a product made from process one, academic manufacturing. And this chart shows anti-sickling hemoglobins as a percentage of endogenous hemoglobin. And the orange here is our payload, hemoglobin F G16D. The light blue is unmodified hemoglobin F. And the dark blue is hemoglobin A2, also an anti-sickling hemoglobin. And the key thing that I will say here is over 36 months of follow-up, this patient has had stable hemoglobin F and completely stable payload, hemoglobin F G16D. So with only reduced intensity conditioning and our vector, this patient has had stable payload now for 36 months. The second patient has had stable overall levels of anti-sickling hemoglobin, and they're still benefiting from this. They've had stable levels of hemoglobin F. So both these things combined are truly what impact the patient's phenotype, and you'll see that in their VOE reductions. However, this patient did not engraft as well. So you can see their hemoglobin F G16D has plateaued at a lower level. And we do understand what happened to this patient. This patient had renal hyperfiltration. They had a glomerular filtration rate of 200. So their kidneys were working too much. And because melphalan is a renally excreted therapy, they excreted too much of their melphalan, and they had a subtherapeutic exposure, a subtherapeutic area under the curve of melphalan. So in the future, for patients with renal hyperfiltration, we will be incorporating renal adjustments to updose their dose of melphalan in the event that they have renal hyperfiltration. And finally, patient 3 is our first patient treated with process 2, our newer manufacturing process. And we're quite pleased with the results from patient 3. So at 10 months, they have greater than 38% hemoglobin F, so well above this 30% threshold that in the literature seems to be required for complete symptom resolution. They started out anemic on chronic transfusions, and now they have a normal hemoglobin completely off transfusions. And they also have pancellular distribution of hemoglobin F. So let's dig in a little more into the data demonstrating the changes between process 1 and process 2. So the drug product vector copy number, again, that's how much vector is in each cell of the drug product. For patients 1 and 2, it was rather modest, 0.2 and 0.47. We still had significant clinical response with these modest vector copy numbers because of the potency of our vector. However, we wanted to get this VCN even higher. And so with those changes, we got the product VCN to 1. In the peripheral blood out at 9 months, patient 3 still had much higher vector copy number at 9 months than either of the first 2 patients. And that has carried through into clinical benefit as well. So the payload, hemoglobin F G16D for patient 3 at month 10. Now we're comparing all of these patients, 1, 2, and 3 at month 10 here. And you can see at that same time point, patient 3 had a markedly higher payload as a percentage and in absolute terms. And their total hemoglobin F, also at 10 months, was the highest, both in percent and in absolute terms, in grams per deciliter, was more than twice as high as the first 2 patients. In terms of the distribution of their hemoglobin F, so percent F cells is the percentage of red blood cells that have hemoglobin F in them, and you want as broad of a distribution as possible to protect as many red blood cells as possible. So at 6 months, patient 3 had 71% F cells, markedly higher than the first 2 patients, and they had 92% F reticulocytes. Reticulocytes are new red blood cells that just came out of the bone marrow. And because of this high level, we expected these F cells to rise. We only check it every 6 months. And indeed, this number has now gone up to 96% at most recent check of 96% F cells. One other marker of how strong the engraftment was is peripheral blood vector copy number. So this is how much vector you're seeing in the patient's peripheral cells over time. And you can see that patient 1 started at a peripheral blood vector copy number of about 0.2, and they've pretty much kept that out now through 36 months. And that matches up with their hemoglobin F G16D payload level. It has stayed stable out for 3 years. Patient 2 declined initially and then has plateaued at a lower level. Again, this patient got a subtherapeutic exposure to melphalan. And patient 3 we're quite pleased with because they started at a peripheral blood vector copy number of about 0.7, and they have stayed at that same level over time. So their pattern looks quite similar to patient 1, stable, just at a markedly higher level. And ultimately, the lab's work is important, but what does it mean to the patient? Well, VOEs are what the patients care about the most because this is their symptoms, this is their suffering. And on the left, you see hospitalized VOEs, and on the right, you see total VOEs, including VOEs where the patient came to the hospital but was not in the hospital. Hospitalized means admitted. And you can see for the first 2 patients, for hospitalized VOEs, 88 to 100%, and for patient 3, there's a 100% reduction, pre-treatment to post-treatment. In total VOEs, the first 2 patients had marked improvements, 86 to 93% reduction in total VOEs. Patient 3 has had a complete resolution of VOEs to date. And then what's ahead for us? As I mentioned before, we are currently dosing patients with process 2, but later this year, we will be rolling out process 3. So that is an additional transduction enhancer. And with that, our goal is to increase our vector copy numbers to the 1 to 2 range. And simultaneously, we are developing our commercial manufacturing process with CDMO partners. And as we're developing that process, we will be further optimizing our transduction conditions. We will be further optimizing our vector to a commercial vector supplier, and we'll be centralizing our commercial cell product manufacturing. And with these changes with the commercial vector and also additional transduction enhancements, our goal is to have a product VCN of 1 to 3 for our pivotal trial. In conclusion, I wanted to show you some data about what this means, what reduced intensity conditioning would mean for physicians. And so we conducted some market research with hematologists and transplanters, and we showed them the product profile for a myeloablative curative gene therapy. And we asked them, of your patients with severe sickle cell disease, what percentage would be eligible for gene therapy? And they said 49%. And so then when we added to that market the ARU 1801 product profile, so potentially curative efficacy, but with the reduced intensity conditioning adverse event profile, both short and long term, um, the population eligible for gene therapy went up to 70%. And in this market, so not only does ARU 1801 expand the size of eligible patients, but it would become the preferred gene therapy in this future market simply because of a superior toxicity profile. So the majority of the patients who actually receive gene therapy, the majority would receive ARU 1801. And there have been several new therapies approved for sickle cell disease, and that is wonderful for patients with sickle cell disease. They need new options. The newest additions are they add solid efficacy, but they are not curative. Patients continue to have events while they're taking these medications. And so the gene therapies, such as CRISPR's gene therapy and Bluebird's, the efficacy looks excellent for these products. The preliminary efficacy, they look like they could completely reduce VOEs in these patients. However, um, these therapies require fully myeloablative chemotherapy and all of the short and potentially long-term AEs that could go along with fully myeloablative chemotherapy. And that is where Aruvant comes in, is that we've demonstrated we can get 36-month durable efficacy. We've demonstrated now with our higher VCNs that we can get curative efficacy, 0 VOEs and hemoglobin F levels at 38%. And our first patient has shown durable engraftment now with stable peripheral blood VCNs and stable payloads now out past 36 months. And so with that, our path forward in 2021 is we'll be completing enrollment of our phase 1/2 trial. We are excited to share additional process 2 data as it comes out later this year. And then we will also be able to share additional data on process 3, on our first patient's dose with process 3 in the second half of this year. And our goal is to initiate a pivotal study. That is what we're preparing for right now in the first half of 2022. And with that, I thank you for your attention, and I look forward to speaking in the future.